So cells, sort of the basic definition, cells combined together or cells living within one another can make complex organisms. So just the sort of basic example of this is there's 30 trillion of them, you, that make you. So 30 trillion cells living together, interacting together, make you. You can separate cells based upon their function. So cells that essentially do very similar things to one another are grouped together based upon tissue. So you can think about this as your liver, your heart, right? They're all doing similar things. Right? Your liver's detoxifying, your heart's pumping blood, your kidneys are filtering, right? And so those are all cells living together, and they all have a very similar function overall. All cells have proteins, DNA, carbohydrates, and membranes. So there's a pretty standard structure to cells, but as you'll see that cells can take on a pretty variable set of functions, and they can also take on a pretty variable set of shapes. And so as we've talked about, there are two distinct types of cells. We have our eukaryotes and our prokaryotes. So last class, we spent all class talking about these prokaryotes, the little guys, the bacteria, the archaea. Remember, they have a very simple cell wall. They're very small with an average of 1 to 10 micrometers in length. Very simple, membrane, DNA, some ribosomes, and that's pretty much it. So it's very, very simple. But depending on which type of eukaryote we're talking about here, we're talking about something that's 10 to potentially 100 times larger, and something that is much, much more complex here on the left. So you can see our eukaryotes, just relative to our prokaryotes, they're just filled with more stuff. And clearly this is something that's just had maybe not more time to evolve, but simply has diverged and went into a much more complex form. And so we're going to talk about all the nitty-gritty pieces here, how they come together, how they work, how you work at the cellular level. And one of the sort of important things about biology, and I've mentioned this before, and I'm going to mention it again, is that structure correlates with function. And so the structure of your cell, both its sort of shape and what types of organelles it has, so all these sort of little names you're seeing here that we'll get into in a moment, its structure and its sort of composition dictate what it can do. So the inner guts of a liver cell look very different than, say, the inner guts of a brain cell. And they also have very different overall shapes. And the same thing here, we can look at an animal cell versus a plant cell. Plants and animals do very different things. And as you can see, they have very different shapes. They have very different sets of organelles. And so structure and function are linked together. That is a very common theme throughout all biology. And I hope you do take that with you when you leave this course. So, but there's sort of the analogy we're going to make today to sort of understanding a cell. It's comparing how our cells work to sort of the whole overall human anatomy. Because we have, a human, we're made up of a dozen or so organs. And each of these organs has a specialized cell, but they're ultimately all contained within sort of our body, right? Sort of that inner, or sort of our outer organ, our skin, right? And our skin sort of maintains and keeps all these organs separate from everything around it. And everything has, again, its own specialized function. And an animal cell and a plant cell is very much the same. It has an outer membrane, and it has all these little organs. And these little organs have been called organelles, or little organs. And each one of these organelles, whether it's a centrosome, a cytoplasm, an endoplasmic reticulum, they'll have their own very specific set of functions, very much like, say, your organs in your body would. So for instance, your kidneys and your liver are really good at detoxifying and getting rid of stuff. Well, you have a lysosome inside your cell, which you can see here, that actually does a very similar function. So there's a lot of parallels here between what's going on in your cell at the cellular level, as well as what's going on at the 38 trillion cell level, that is your sort of overall complex organ system. So we're going to start at the outside, and we're going to start at the membrane, because the membrane is by far the most important part of your cell. Well, from a sort of, maybe not for how your cell functions, but it definitely is the most important part in terms of keeping your cell separate from the inside of your cell, separate from the outside of your cell. And that's a very sort of important thing, right? You could picture, like, if you just removed your skin, right, that would just be a pretty, not only just a frightening thing, but it would also be a pretty detrimental thing to you. And the same thing if you remove the membrane of your cell would basically just die. And so the membrane is an extremely important part of the cell. But what you'll notice is the membrane is not all that complex, right? It kind of looks like a sandwich. It's got some little nubbins hanging through it. It's got some sugars or carbohydrates hanging off of it. And that's more or less it. It's a pretty simple system, but it has some very important functions, and it has some important properties that we're going to discuss today. So the question we can ask is, why do we care? And the important thing is it creates this homeostasis, this idea that the inside of you is distinct from the outside of you. Again, the analogy of your skin, right? Your skin keeps you separate from the outside world. And that's an important thing for how your body functions, for preventing infections, and so on and so forth, right? And so that, keeping that homeostasis, keeping that internal inside conditions is an important thing to do, not only at the cellular level, but at the sort of the whole organ system level as well. Like your skin, it provides the first barrier of protection, and like your skin, it allows and limits the movement of things inside and outside your body. So it really limits what can physically go into your cells, right? So if you think about your skin, say you go swimming in the pool and your fingers get all swollen up because you've been in the pool for a long time because they're all saturated with water. But then you could say put them in another liquid, say, I don't know, something weird like mercury, right? Your skin wouldn't actually let that mercury pass through, but it lets the water pass through, because it has what we're going to talk about as selective permeability. And that's the same thing your membranes have as well. And so just sort of bringing this back to a few lectures ago, I hope you guys all remember these mycelia, these protocells, essentially, like these membranes that we're forming. This sort of concept of inside versus outside, remember, was also important not just for modern day life, but it was actually important for early life. And that's in particular back in sort of olden times. I say olden times as in like billions of years ago, not like a couple hundred years ago. Was important back then because of how tough life was on Earth, and sort of keeping that balance between terrible outside and nice inside was an important thing early on as well. But membranes are composed of lipids, and you guys have all heard of lipids before in some shape or form. You've heard of fats, you've heard of oils, you've heard of all sorts of different things that can be considered a lipid. But in particular, the lipids that make up your sort of overall cellular membrane are what we call phospholipids. And we'll get into the structure of those first, but let's just sort of mention a few little terminologies about lipids in general. So most lipids typically have a hydrophobic and hydrophilic part, with hydrophobic meaning fear of water and hydrophilic meaning love of water. So it has this pretty strong dichotomy between a part that likes water and a part that hates water. And when we're thinking about our membranes, and we're thinking about fats in general, when we think about a fat, we're thinking simply about something that is chock full of carbons and hydrogens. This is similar to, you can actually look at, like, say, the chemical formula of gasoline, octane, and octane looks very similar to this. And so things that are sort of chock full of hydrogens and carbons are called hydrocarbons. But what's sort of distinctive about our fatty groups is, and our fatty acids in general, they have these what are called carboxyl groups tacked onto the end typically, or tacked on somewhere along the way here. And this is what makes a fatty acid distinct, or a fat distinct, from something that's just, say, a hydrocarbon, which again, hydrocarbon just being a chain of carbons and hydrogens. And fatty acids can be saturated or unsaturated. So I'm sure you all have heard this before when you looked at the warning label, or not the warning labels, but the ingredient labels on your food. I'm sure you've noticed that there's a label on your food for fat, unsaturated fat, and saturated fat. And the key difference here is just this double bond here. That's all that really is the difference between saturated and unsaturated fat. By adding this double bond, you remove some hydrogens, as you can see, and it essentially kinks the fat. And this kink in the fat can actually make the fat more likely to get stuck sort of inside you as it sort of moves around, which is typically why I would think of unsaturated fats being not so good. But that's our fats. And just as a note, the way the fats look at the sort of the chemical level, so the string of carbons and hydrogens, and the presence and the absence of a carboxyl group, or the presence and absence of double bonds, really sort of gives the properties of these different different oils and fats and lipids that we know of. And so depending on how you change the orientation of all this stuff at the chemical level, you can change the behavior of this. So for instance, we have saturated fats like butter, which is the best of all saturated fats. You can see that it's completely, so anytime you see like a sort of a line like this, it's just a bunch of carbons and hydrogen. So this is just, it's carbohydrate, it's a hydrocarbon tail here. What you're seeing is that this is a saturated fat, and it's solid at room temperature. That's in contrast to something like safflower oil or sunflower oil. You can see it is unsaturated, meaning it has those double bonds, and you can see it's liquid at room temperature. And then you can look at something like beeswax, which is unsaturated. I'm sorry, it's a saturated fat here, but it's got very long hydrocarbon tails. But what you'll notice is that the sort of overall structure of these different hydrocarbons, these different fats, give them very, very different properties, right? If you try to make a membrane out of this out of this beeswax here, right, that would do a much better job than, say, trying to make a membrane out of this safflower oil here. So it very much like your proteins, where you change the sort of underlying ingredients here, you can change the overall properties of a fat or a lipid by simply changing how the hydrocarbon chains are oriented. That's a hard word to say this morning, I apologize. But more importantly, thinking about your membranes, they're a very specific type of lipids, and these lipids again are phospholipids. And so the main characteristic that we look here is, again, that hydrophobic tail, meaning it hates water, and that hydrophilic head. And now that hydrophilic head is comprised of a phosphate group, as you'll see that PO4. That should be familiar from ATP at this point. In addition, you have what's called glycerol, and it's just a carbon and oxygen compound. And all of that hydrophilic head is attached to two tails, one a saturated fatty acid, and the other being an unsaturated fatty acid. And so this sort of overall phospholipid structure is basically the way we make our membranes. Bacteria will make their membranes in the exact same way. Archaea have a slight variation on this formula, but they do it pretty much the exact same way. And that, not to sort of oversimplify membranes, but there are some sort of funky guys out there that do things slightly different, but by and large, pretty much all of life on Earth does this sort of overall function. Hydrophilic head, hydrophobic tail. And one of the reasons we sort of believe this was something that's really shared across so many parts of life is this was something that actually evolved potentially early on. And I'll get into this in a second. But the idea being is if you have these hydrophilic heads and these hydrophobic tails, they will self-organize themselves, meaning that the hydrophobic tails will try to shelter themselves from water if they're in a solution of water, and the hydrophilic heads will sort of orient themselves outward. So they'll naturally, just based upon the sort of the properties here, will essentially block the phobicity with the philicity here. That was a weird way to say it, but that's what's happening here, and form natural circles like this. However, more commonly when we're thinking about membranes, the most common layer we actually see this forming is not sort of this circular way, because as I mentioned, when you think about your cells, you're sort of just kind of a bag of water, right? And if this inside was a bag of water too, that wouldn't be particularly helpful for all these hydrophobic tails. However, most commonly what we see is they form what's called a lipid bilayer, where we have two layers of these fatty acids, and when you have the tails facing the inside. So essentially you make a fatty acid sandwich. But again, you have an orientation of hydrophilic heads at the outside and hydrophobic tails on the inside, forming this bilayer. And this bilayer, this sandwich, is the sort of the way we think about pretty much the majority of cellular membranes across the tree of life. And this sort of overall shape should remind you of something from our Origins of Life class in these amphiphilic molecules. And so these molecules, again, self-orient themselves, as I mentioned, but this is also a candidate for the way early membranes on the planet Earth look like. And remember, we could find these asteroids that can be synthesized chemically. And so this sort of early template, we think, led to sort of this much more complex, sort of double-tailed bilayer that we see in modern time. So that's our phospholipids. They make up the vast majority of what's going on in your membranes. The next up are steroids, and steroids are just a class of lipids, and they're typically embedded inside animal cells. And it's not that common, but you can find them in other organisms outside of animals, but you generally only find them in animals. And so a steroid is really sort of a very simple thing. It's got a polar and hydrophilic head, this OH, this hydroxyl group. It's got a bunch of rings, and these rings are carbon. and these are hydrophobic. And then it's got, at the end of it, it's got an isoprenoid tail, and that's sort of the basic structure of a steroid. And steroids essentially are embedded inside your animal cell, and we'll talk about sort of their function in a minute as we sort of progress through sort of the things you stick inside that phospholipid bilayer. Next up is proteins, and these proteins can be one of two classes. They can be integral or transmembrane, and proteins are really strongly involved in cell-to-cell communication, transport, and sensing inside your cell. Remember, proteins pretty much do everything inside you, so you can imagine that a protein that's embedded in your membrane has a lot of function to it. And so, as I mentioned, proteins can be integral or transmembrane. Something that is transmembrane essentially spans the entire thing, meaning it connects outside from inside. And proteins that are typically transmembrane like this, they act as channels, allowing a cell to pump things from outside to inside or inside to outside. So that's our transmembrane proteins. Integral proteins essentially just embed themselves on one side of the membrane. So they essentially don't cross the membrane sort of at all. They sort of just, they essentially have one side sticking out, say either on outside or inside, and then the rest of it is just embedded in. And there's really no trans sort of properties to it like you would see here. Sometimes they'll have outside proteins attached to them, but when we're thinking of an integral protein, it's really something that's just sort of hanging out in the middle of the membrane, only really involved on sort of doing things on one side of the cell versus the other. Next up is carbohydrates, and we typically think of these as sugars, and these are typically embedded in the membrane in some capacity. So sometimes they'll be embedded directly into the phospholipid bilayer, or more commonly, they'll be embedded inside of a protein. And the idea being is that we commonly use these in our own cells to essentially say like, hey, I'm a liver cell, or say, hey, I'm a brain cell, right? So they're really commonly used for giving cells information about one another, saying, hey, I do this, this is what I do. And so all these sort of aspects of what you can make a membrane out of, sort of starting at the sort of the bilayer and then extending out to proteins, steroids, as well as carbohydrates, give membrane different properties. And membranes, like most things, have different properties depending on a sort of a wide range of conditions. And so the most common thing that affects a membrane is what you have embedded in there, so how much proteins or steroids you have in there. Another really common thing that affects membranes is temperature. Third, we have the pH, and then finally, what type of lipids you have. And so remember, you can modify lipids by changing them from saturated to unsaturated, which, as you notice, gives them some very different properties. And you can sort of think about the properties of a membrane simply as being important, because a membrane is not static, right? It's not like a wall, right? It's not a hard surface that prevents things from going in and out. It has to be a surface that's permeable. It has to be a surface that's flexible, right? If you think about your skin, right? You press down on your skin, there's some give to your skin, right? There's that flexibility to it. And your membrane has to have an overall level of flexibility. And one of the ways it does this is by the sort of the basic fact that the phospholipids that make up your sort of your bilayer are always in constant lateral motion. And so essentially you could think of what makes your cells, that membrane, those lipids are always in constant motion. Always sort of moving back and forth. They never flip sides. Well, they can, but they very rarely do. But they're generally just kind of moving along. It's basically like you can think of like a crowd of people, and people are moving around. That's basically the way sort of lipids are. They're just sort of all constantly moving. It's a fluid environment. It's not static. Again, it's not like a rigid wall. It's very much fluid. It allows things to move both in and out. So membranes have to have these properties to allow things to move in and out. And the first way they allow these membranes to allow things to move in and out is by these phospholipids themselves being in constant motion. And so by having different properties, it gives these membranes the capacity to transport things across. And so, as I mentioned, the constant motion of the lipids is the first way you do this. But you overall do this to give your membranes the property to move things. And so the way your membranes move things is primarily by two different means. The first being passive transport. The idea being passive transport is that these molecules or these proteins or whatever you're talking about, they move across the membrane without any help. They just sort of do it by themselves. And the next step after that is active transport, where a cell physically grabs onto something and pulls it inside of its cell, or physically expels something from the cell. And when we're thinking about how things move by passive transport, we're thinking primarily of diffusion. And so when we're thinking about diffusion, this is how everything moves but water. And we'll talk about water in a second, because water is a special boy. But everything on the planet outside of water moves by this process of diffusion. And the idea being it moves from high concentrations to low concentrations. So you can think about if we were in the classroom, right? And I stacked all of you up on one side of the classroom and I stood on the other side. Well, now there's a gradient where there's very little people on one side where I'm standing, and there's a lot of people on the other side where you all are standing. The idea being diffusion will naturally, just through this gradient, will move some of you from the very concentrated size over to me, again, naturally, until we're in equal proportions on both sides of the room. That's what diffusion does. So let's just look at an example of this. So we have some sort of aqueous solution here where we have two chemicals dissolved in, purple and white, and we have some sort of permeable membrane here in the middle. You can think of this permeable membrane being analogous to the lipid bilayer in your cell. And the idea being is there's a concentration gradient. There's no white here, there's no purple here, right? What diffusion does is it mixes those two compounds. The idea being is there's a gradient. So the purple moves across, the white moves across, and eventually you reach what's called equilibrium, where there's equal numbers of purple and white on both sides of this membrane. And that's what diffusion does. Now, and now I want you to think sort of this sort of movement process stops. Just as a note, even when a cell reaches equilibrium, there's constant back and forth of molecules. Just as a note, like it's just the way things work. I don't want you to think like, this is just static, it just stays in equilibrium. It really doesn't. Things are always moving. Remember, for something to have temperature, right, it's always about the movement of molecules. So there is always movement back and forth, even once you reach equilibrium. But they do stay in equal proportions, so just as a note. So that's diffusion. Again, high to low. Pretty straightforward, which is nice. This is how everything but water moves. Everything but water. And water, as you all learned, is a weird boy. And water moves not by diffusion, but water moves by osmosis. And water is weird. It moves from where concentrations of not water are high, but where concentrations of solute are high to concentrations of solute. I'm sorry. Well, that's a mistake on the slide. Water moves from where concentrations of solutes are low to where concentrations of solutes are high. Sorry, that's a mistake on my part. I can't believe I missed that. But the idea being is if we have, again, this lipid bilayer, where we have low concentrations of something dissolved in water on one side and high concentrations of water dissolves on another side, what water does is it moves by osmosis from this side where there's low concentrations to this side where there's high concentrations. And so water is different. Nothing else in the planet moves like water does. And this is all due to those funky properties of water that I've mentioned in the very first class together. And the idea being is that water will continuously move from low to high until the concentration of this, whatever's dissolved in here. So when something is dissolved in water, it's called a solute. And when the concentration of the solute is equal on both sides. And so it's just a funky thing water does, because it's at the chemical level, what's really happening is the water's attaching onto these solutes inside these, causing them to move across the layer. That's what's really happening. But again, it's all hydrogen bonding here. But the idea being is it will move, and you can actually do this experiment if you had sort of this setup, and you would see this exact same thing. But again, what water is doing here is it's equaling the concentrations. It's equaling the idea being if I have five milliliters here, and if I originally started with say 5 milliliters of water on both sides, the idea being is that water would move from this side to this side. This would expand the amount of water here, but what it would do is the concentration of the solute here would then be equal to the concentration of the solute here. That's what water is doing. Sort of real world, sort of thinking about what sort of how we describe these situations. If we have what's called a hypertonic solution, the idea being is there's we're thinking about this in terms of a cell, right? If we have higher concentrations of something outside of the cell than inside the cell, water will move out of the cell from the low concentration inside the cell outside of the cell, thus causing the cell to shrink. In a hypotonic solution where we have higher concentrations of things inside the cell than outside of the cell, water will move into the cell from outside of the cell, causing the cell to swell. And then finally, in what we call an isotonic solution, there will be no net movement of water because the concentrations will be equal both inside the cell and outside the cell. And I know that was complicated, but the idea being is this what we think about in a real world application. So if we have our red blood cells here, I'm sure you guys have all seen these before. In an isotonic solution where concentrations are equal on the inside and out, there will be no net movement of water and the cells will not do anything at all. In a hypertonic solution, say our cells are in a really, really salty water solution, what will happen is water will essentially leave our cells to go to the really salty solution, causing our cells to shrivel up and die. And then finally, in that hypotonic solution where we have higher concentration inside the cell versus outside the cell, water will essentially flush into our cell. And if it pushes enough water inside of our cells will eventually explode, or what's called lysing. So this happens, all the time, sort of naturally speaking, and can happen inside your body. But this is what happens when water moves. And again, water is always trying to go to where things are in the highest concentration. And this is all due to those oh-so-fun hydrogen bonds that water has. And so as I mentioned, membranes separate the in from the out, and they have what's called selective permeability, meaning they only let certain things in and out. And this permeability is sort of tied to a couple different things. And so this permeability is tied to size and sort of whether or not something carries a charge. And so we have a hypothetical membrane here, our phospholipid bilayer, and we have a permeability scale. And this is in how fast things move, so centimeters per second. You don't need to think about the units. You don't really need to think about the units or the numbers here. This is just a scale. Just think about it as this has very low permeability, meaning it doesn't move very fast across membranes. And this has very high permeability, meaning it moves very fast across membranes. So the idea being is that things that are gases move very quickly inside and outside of your cell. And that's a very important thing, right? If you breathed in oxygen and you're creating CO2 by respiration, you want that to be able to leave your cells in an orderly fashion or come into your cells in an orderly fashion, just because CO2, if it builds up, would be toxic, right? So gases move pretty easy. Things that are very small, like water or glycerol, which again is that small carbon compound that makes up some of your phospholipids, that's very small. It doesn't carry a charge, and those things will move relatively easily, but not as well as, say, gases will. Now, moving sort of to larger things that don't carry any charge, such as sugars, they will move through your membranes, but they do it very slowly. It's very hard for big things like a sugar or even something bigger like a starch to move through your membrane. And then finally, the things that have the lowest permeability or the capacity to move through these membranes is charged things. Things that are charged like potassium or sodium, they don't move through your membranes at all. They essentially get repelled by your membrane. And so there's this overall permeability scale here, and it's all about how big you are and how much of a charge you carry on yourself. And so that's how we that's sort of how your membrane is selective in what it lets through or not. And just to sort of reiterate sort of what we talked about for these membranes, the membranes are created, are created by a phospholipid bilayer. And remember, they form this way because they have a hydrophobic tail, which means they hate water, which forms the inside of the membrane sandwich, and a hydrophobic head, meaning it loves water, forming the outside and the inside part of your membrane. And now your membranes, again, they separate the in from the out. And you remember, your inside of your membrane is essentially a bag of water. The outside of your membranes, again, is also essentially a bag of water. Inside your membranes, you can embed steroids, you can embed carbohydrates, and you can embed proteins. In addition, animals can also embed cholesterol inside of our membranes. Plants do not do this, so you can't get cholesterol from plants, which is nice. But what this does is very much like steroids. It actually improves the membrane flexibility, allows the membrane to sort of maintain its sort of capacity to wiggle. In addition, remember those carbohydrates allow, essentially provide a fingerprint for the cell, so it can be recognized by other cells. It can be recognized by molecules that are coming through your, say, your bloodstream. And then finally, you have your proteins, which are doing a lot of stuff. So you can have proteins that are participating in reactions inside or outside your cell, or you can have proteins that are transporting things from inside and outside of your cell, doing that process of active transport. So that's the membrane. That's the sort of that's the outer part of your cells. It's really important for maintaining you from the environment. And I know that wasn't a very exciting thing to talk about, but the membrane is an important aspect of your cellular biology. Next up, and probably more exciting, is the nucleus, which forms essentially the brain of your cell. This is where the vast majority of your cell's DNA is housed. And I say the vast majority is because, as we'll talk about when we talk about chloroplasts and mitochondria, two other organelles, they do have their own separate genomes. And so just as a note, just as a foreshadow, but the vast majority of your cell's DNA is actually housed in your nucleus, which is why we call it the brain. And remember, thinking about what DNA is for, remember the DNA is the storage medium for all biological information. And it also allows your body to make, your cells to make copies of the DNA, in particular the genes, to make proteins. Remember, proteins do everything. And just as a final note about the nucleus, it is encapsulated by a membrane. As you'll notice, everything going forward, everything inside of a eukaryotic cell is encapsulated in some membrane. And so remember, just as a reminder, the DNA is read to make RNA, that messenger RNA. And that messenger RNA is then sent out to make proteins in the ribosomes. And so your DNA has what are called nuclear pores, which allows things to move in and out. In particular, allows the RNA to move in and out. Sometimes DNA will move in and out, but primarily it allows RNA to essentially escape. You can think of this like a, like a wiffle ball, almost like. That's sort of what the overall shape of, in the sort of the overall look of a nucleus is. It's like a wiffle ball. And sort of smack dab in the middle is your nucleus. And smack dab in the middle of your, and sort of spread out is your DNA. So there's a question in chat. It says, do the gases and glucose we just mentioned run through the protein pathway? So the gases themselves will just diffuse straight through the lipid bilayer. In terms of the physical sugars and stuff like that, if they're very large, they have to be moved through a protein. If they are small, they can, like say a glucose molecule can potentially diffuse directly through the membrane, but most sugars have to go through a protein pore in the membrane. So they'd have to go through one of these channels by and large. But gases typically, unless it's like a chlorine gas, will go right through the membrane. So any gas that's not charged, water, will typically move right through the membrane. Things that are big, sugars in particular, they need some channel. They need one of those proteins. So again, in terms of your nucleus, it's this is where you house your DNA. It's sort of spilled in there like a bowl of spaghetti. And another sort of aspect of your nucleus is what we call the nucleolus. And the nucleolus is essentially the site of both ribosomal RNA synthesis as well as ribosome synthesis. And just as a remember, the ribosomes are the site of protein synthesis. And we typically think of the nucleolus, especially in recent times, because actually, funny enough, when I was learning this stuff in high school biology, so in the early 2000s, people were still sort of perplexed as to what the nucleolus did, which I think is a kind of interesting thing. But now we know the nucleolus is the sort of the site for ribosomal RNA synthesis, and it's essentially a control center for stress. And so there's all sorts of stresses your cells can face on an everyday basis. They can think of something like osmotic stress, which is water stress. They can think of hypoxia, which is oxygen stress, nutrient deprivation, heat shock, as well as things like metabolic stress. And so your cells experience these on an everyday basis. And depending on what type of cell it is, it experiences it much more common. And a way a cell can respond to this, as we'll talk about, I believe, next class, is by changing how it transcribes its genes. Because remember, all a cell can really do is take its DNA, transcribe it, and make proteins. And so thinking about that, thinking about how a cell essentially responds to stress, it's really controlled by the nucleolus, this really tiny little organelle that essentially directs the cell to make more ribosomes, which by making more ribosomes allows you to make more protein, which allows you to then respond to stress. So the nucleus has its own little special function related to how a cell responds to stress. And so certain groups of cells inside your body, say like your skin cells, the nucleolus is doing a lot all the time. That's in contrast to, say, something like maybe like your nerve cells, which don't really experience a whole lot of stress on a day-to-day basis. They have electricity flowing through them and stuff like that, but not at the level of stress that, say, your skin or your liver would experience on a day-to-day basis. And so sort of going back to the central dogma of biology, remember the DNA, RNA, protein, right? Transcription from DNA makes RNA, and then once we make that RNA, we translate it into the proteins. And so that's that's the sort of flow of information. That's the way a nucleolus will respond to a stress, right? It takes the DNA, it says, okay, here's the stress, here's the DNA, here's the genes I need. It transcribes those genes and sends those to make protein in the ribosomes, or it essentially directs the cell to make more ribosomes to make more proteins. That's the sort of the flow of information that the nucleolus works with. Next up is the endoplasmic reticulum, or the ER, and this is the site of protein and lipid synthesis. And this essentially is something that can be rough or smooth. Just to answer your question in the chat, Peng, yes, the cell is just like a factory, and DNA is the blueprint. That's basically your cells are. You're basically just an assembly line. As you'll see next class, there's really an assembly line function that goes from DNA to RNA to ribosome to make protein, and then sort of this assembly line fashion to get it where it needs to go. You'll see it's kind of like Amazon. It's just not as like sad as Amazon is with all the terrible things Amazon does to their workers on a daily basis. But you'll see it's really like it is like a mini factory. So it's a pretty good analogy. So again, endoplasmic reticulum, the site of protein and lipid synthesis. So as I mentioned, there are two endoplasmic reticulums, the first being the rough endoplasmic reticulum. And this endoplasmic reticulum is rough simply because it is covered in ribosomes. It's covered in these little pimples that are ribosomes. And so this is what it looks like in a cartoon fashion. This is what it looks like under an electron microscope. So you can see the size we're talking about here, 200 nanometers is the scale bar here. So it's something, we're still at a very small scale here. I don't want you to think like we're in a eukaryotic cell, things are big. Things are still tiny. But what you'll notice about the endoplasmic reticulum is that essentially it's two things. First, it sort of engulfs the nucleus. It sort of directly surrounds it. And the second thing you'll notice, it's essentially a series of folds, right? It looks like you took some deli meats and stacked them on top of each other. At least that's what it looks like to me. There's probably a better analogy for that. But you see, it's just a series of membranes. And essentially, these membranes are covered in these ribosomes, these sort of like pimple-like structures. And the closest one to the nucleus is our endoplasmic reticulum. That's rough. Outside of the rough endoplasmic reticulum, we have the smooth endoplasmic reticulum, which is very similar to the rough, simply just doesn't have any of those ribosomes on it. So the idea being is, while most of the protein synthesis in your cell goes on in the rough endoplasmic reticulum, once you make those proteins, if you're going to make lipids from those proteins, most of that lipid production happens inside the smooth endoplasmic reticulum. And so the idea being is that if we're thinking about the flow of information, we have DNA transcribed to RNA. That RNA is then commonly sent to the rough endoplasmic reticulum, where one of those ribosomes will make that protein. Well, once you make the protein, how do you get it somewhere inside your cell? And the answer to that is going to be the Golgi apparatus or the Golgi bodies. So once you make your protein, or once you've taken your protein and sent it to the smooth endoplasmic reticulum to make a lipid, you got to get it somewhere. You got to ship it somewhere. And the Golgi apparatus is your man. It is like the Postal Service, even though it's struggling a little bit right now. It operates in rain, snow, sleet, hail, and shine. It really does work all day long, and it packages and processes essentially everything your cell makes. And so this is what your Golgi apparatus looks like. You typically have multiple spread across your cell, and very much like your endoplasmic reticulum, they're essentially a series of folds. They're very small. So again, 100 nanometers is the scale here. Very, very small. What you'll notice is they're oriented, both attached to your endoplasmic reticulum. So this one's attached to the smooth, this one's attached to the rough. And depending on what type of cell you are, so for instance, if you're a liver cell, you have many, many more Golgi apparatuses. In contrast, if you're, say, something like a skin cell, you have very little Golgi apparatus. So it just depends. Some cells have a lot of Golgi apparatus, some have very few Golgi apparatus. And one thing I want you to keep in mind here is, again, this is just a series of folds. Essentially, this is all membranes. This is just a series of folds. But one thing the Golgi apparatus has, unlike the other sort of organelles that we're talking about, it has two different faces, and these faces behave differently. And so it has what we call a cis side and a trans side. The cis side always faces the endoplasmic reticulum, whether it's rough or smooth, and then it has a trans side. And the trans side faces the outer plasma membrane. So just as a note, this isn't as important today, but just keep this in mind for tomorrow. We sort of talk about how you put all these things together. So the idea being there's a cis side and there's a trans side. There's directionality here. And just as sort of a foreshadow, if we say got a lipid from the smooth endoplasmic reticulum, it would move from the cis side of the Golgi apparatus to the trans side of the Golgi apparatus. And once it went there, it would then be packaged and shipped to wherever it wanted to go. But there is a directionality here. Unlike the other organelles we really talked about. There's no directionality in those. I mean, there's inside, outside, but not a directionality. There's no definitive way information flows in these systems, but there is on the Golgi apparatus. Next up is the lysosome, and this is the garbage disposal of the cell. It recycles old, used, or extra materials inside your cell. It also is a site of detoxification. It does a whole lot of different things. And so this is what a lysosome looks like. They're just these little circular membrane-bound things. They're most commonly depicted as yellow. And I always wondered why they were always depicted as yellow. And I learned recently that the most common color of waste produced when you decompose things inside your cell is actually yellow. So just a fun fact. And so these lysosomes, again, they're a site of recycling. So the idea being is if you say you have some RNA and you want to get rid of it because you don't need to make those proteins anymore, you send it to the lysosome. Say you have some proteins you want to get rid of, you send it to the lysosomes. If you want to have some lipids you want to get rid of, send it to the lysosome. The idea the lysosome releases a couple different things. The most common thing it will release is hydrogen peroxide, and that will essentially dissolve what you're trying to get rid of. It also releases other acids and things like that to break down parts so that essentially it can do one of two things. If it's a lysosome sort of close to your nucleus, like say this one is, or if it was say over here, it will essentially recycle those back into your cell, reusing the materials. And you know, I hope it's sort of kind of like intuitive that a cell might want to recycle its things because, again, there's always a limited amount of materials out there for a cell to use, even something as big as us. So lysosomes can be used for recycling, but if you have a lysosome that's really close to the membrane or sometimes even touching the membrane of your cell, it'll actually just spit that stuff out. It'll essentially eject it out into the outside world. And this will, if you're thinking about a human cell, this lysosome ejects stuff out into the bloodstream, and the bloodstream will then carry it to essentially the liver and the kidneys to essentially process it and package it for essentially excretion, right? Urine or feces. So lysosomes are how your cells dump things into your overall grander garbage disposal system of your body. So analogous to a lysosome, inside of a plant, we have what are called vacuoles, and these are found only in plants and fungi. Not all fungi have vacuoles, but all plants have vacuoles. And vacuoles, like a lysosome, will digest old material. They'll recycle things. But the important things that vacuoles do is actually store materials. And in particular, vacuoles will store water. That's the most important thing a vacuole does for a plant. So what you notice is that we have a vacuole. It looks like just kind of like a mini lake inside of our cell. You'll notice that the plant cells are much more rigid in shape. They look like, this one looks like a pentagon, essentially. No, I'm sorry, this is a hexagon, not a pentagon. But if we look at an electron micrograph, you'll notice that the vacuole is huge. And that's how most vacuoles are. Most vacuoles take up at least 60% of the volume of the cell of a plant. They can take upwards of 90% of the volume. But what you'll notice is that this vacuole is depicted as being like a mini ocean. And the most common thing a vacuole is used for is to store water. And this is an important thing for plants. And for those of you that ever tried to grow anything, you'll know, and have failed at growing those things, you'll know this, and this will sort of hit you in your heart. But when a plant's vacuole is full of water, it causes the plant to get what we call turgor pressure, essentially forcing the cell to get bigger, to get tighter. And what it does, when you have a nice, full, and stiff plant, it allows the plant to be rigid, to stand up. And they eventually will die, but they first start to wilt. So this pressure inside the vacuole, this turgor pressure, gives a plant the capacity to stand up, to hold itself up. And the absence of that causes a plant to wilt. So for those of you that are like my wife and don't have a green thumb, and your plants look like this, pretty commonly, it's usually because they don't have enough water inside them. So, fun facts. Next up is the mitochondria, and this is what we call the powerhouse of the cell. I learned recently that this is mitochondria being the powerhouse of the cell is a meme, like, you know, one of those funny cartoons. So I didn't know that till recently, so I think that's kind of interesting. But it is the powerhouse of the cell because this is the site of cellular respiration. And so a mitochondria is something we find in both plants and animals. And remember, cellular respiration is taking chemical energy in the form of sugars, fats, proteins, and making ATP of it, right? Because ATP, adenosine triphosphate, is the energy currency of your cell. And sort of one of the interesting things about mitochondria is they actually have their own genome. So let's look at a mitochondria or a mitochondrion, interchangeable words here. You'll notice, like all the other organelles we've talked about, it is bound by an outer layer. But also sort of like the endoplasmic reticulum, it is a series of membranes. And so these little membranes on the inside, so these, you think about these sort of, not the blue parts, but sort of the red parts in this diagram. Those are what we call the cristae. And so these are membranes. And then we have what's called the matrix. And the matrix is not like the movies, but like it's just empty space. And the idea being is it uses this series of sort of membranes and empty space to essentially perform that cellular respiration. We didn't really get into sort of the nitty-gritties of cellular respiration, but cellular respiration requires membranes to sort of create a gradient. And by having many, many membranes embedded inside the mitochondria, it creates lots of sites for respiration to occur. So, next up, actually one other thing I want to mention about the mitochondria that I think is an important thing is that it has a membrane, but it has two membranes. So the mitochondria is special. Everything else in your cell has just one membrane. The mitochondria actually has two membranes. And the reason it has two membranes is because respiration is a sort of a nasty process. You produce all sorts of nasty chemicals as you perform respiration, including things like formaldehyde, reactive oxygen species like, for instance, hydrogen peroxide. So you produce all these nasty things that are toxic to your cells. And your mitochondria has this, again, this extra layer to prevent those things from getting out. Because if something like really toxic, like say formaldehyde or hydrogen peroxide got out, it could potentially damage your other organelles or potentially damage your DNA, which would be very bad for your cells. So the mitochondria is, again, it is a place where lots of important things happens, but it can again be a place where very bad things are produced. So it has this double membrane to essentially protect it from the rest of your cell. So really keeps things isolated. The chloroplasts are something that is found only in plants, and these, as well as other photosynthetic organisms, including algae and other types of photosynthetic eukaryotes. And as we've talked about, this is the site of photosynthesis, converting light energy to chemical energy, right? So they're fixing CO2 to make sugars. And very much like your mitochondria, it has its own genome. So let's look at these chloroplasts again. We've seen these before when we talked about energy. So it has these what we call these glycoloids, and these are these little stacks of, like, you can think of them like Oreos, and these are the glycoloids. It has inner membranes and stuff like that. And each of these individual stacks are called granum, and then sort of the inner open space of a chloroplast is called a stroma. So this is what a chloroplast look like. Remember, each of these stacks of glycoloids, or granum, whatever you want to call them, they are, again, this is where chlorophyll is housed. And remember, chlorophyll is the light capturing pigment. And so you'll notice this is green. And remember, chlorophyll is green because chlorophyll absorbs red and blue light and reflects green light back up to your eye, which is why they appear green. But what you'll notice is that in this particular case, this plant cell has 4 chloroplasts. And the amount of chloroplasts per plant varies. And so there are some plants that will have hundreds of chloroplasts in a single cell, while there'll be others that have maybe 1 or 2 or 4 or 25. There's a sort of a variability. You can have 1, you can have hundreds. And so it just depends on the plant as a whole. But again, this is the site of photosynthesis. This is why it appears green. And sort of an overall, sort of a last note here between the plants and the animals before we sort of wrap up this little talk about this is they're very similar, right? You'll notice that between a plant cell and an animal cell, they have a nucleus, they have a nucleolus, they have ribosomes, they have mitochondria, they have endoplasmic reticulum, right? There's lots of similarities here, but there is some differences that do occur that are found only in one versus the other. And so we've talked about a couple of these already. First being the chloroplast and the vacuole, only being found in plant cells. Plant cells also have one other specific type of organelle, which is called their cell wall. It's a really thick, rigid outer layer that you found in all plant cells that you do not find on animal cells. And this really rigid outer cell wall helps maintain, essentially absorb the force of that turgor pressure from a vacuole being filled with water, which allows the plants to essentially swell. Remember, that swelling is important for plants standing up straight. But what you'll notice is in this diagram here, again, that vacuole is monstrous, right? It takes up about 90% of the cell's volume. So just as a note. That's pretty much it in terms of what the plant has by itself. One thing that animal cells have are what are called centrioles, and these will become more important in a few classes when we talk about how cells divide, a process called mitosis. But these centrioles, which you only find in animal cells, are related to a process of cellular division inside of eukaryotes. So if you want a sort of a nice little nerdy wrap up of what we all talked about, sort of talking about the structure and function of all these different organelles, this is a nice little YouTube video. I think it's about 8 minutes long if you want to sort of review it. And it basically goes through what I went through in a much shorter period of time. And I'm sure it was much easier to listen to than to listen to me for 55 minutes. But with that, that's going to be the end of class today. We're going to continue this next class. We're talking more about cells. We're going to talk about how they divide. And we're talking about how all this sort of, these different parts of your cell are linked together and how we sort of go from DNA to physical action. So with that, I hope you guys have a good day. If you have any questions, please let me know. If you have any questions about your homework or your exams, again, let me know. More than happy to help you. But remember, don't wait till the last moment because I can't answer your question at 11:30 at night